Hi Leon, so today's lesson, the first one is about DNA and the genome, so it's going to be really quick, me talking. I'll show you the YouTube video, hopefully you can listen to that, and then I've got some questions that you can answer. So the first thing is we're going to do it a little bit backwards, so I'll talk you through first. So in eukaryotic cells, so that is your plants and animals, DNA is stored within the nucleus. In prokaryotic cells, that is found in the plasmid. The main difference is in eukaryotes, we have a double strand of DNA, and it's a double helix shape, so it's two strands twisted around each other, and it forms what we call that double helix. It's like a twisted ladder. In prokaryotes, they only have one strand of DNA. DNA is arranged into chromosomes, so in human cells, this is 46 chromosomes, or you can say 23 pairs. In the gametes, so sperm and egg, there is only 23, so you get half the genetic information. And when they fuse together during fertilization and meiosis, that is when you end up with your 46 in total. Of that chromosome, when you unravel it, because it's like a big ball of wool, when you unravel that shape, that chromosome, you'll end up with that spiral double helix, which we call DNA. A very small section of that DNA code would be called a gene, and they can code for characteristics like skin colour, hair colour, eye colour. They can code for proteins, they can code for hormones like insulin, glucagon. The sort of possibilities are endless. When you change the arrangement of the DNA, so the arrangement of those genes, you then call that a mutation, and that can lead to, in some cases, nothing happening, or it can be a mutation that could cause sort of issues like diseases, like polycystic kidneys, and so on. So what I'd like you to do is I'd like you to write down a definition of what DNA is, a definition of a chromosome, so that is a long strand of DNA, a definition of a gene. Then once you have done that, you can press play again, and hopefully I will show you a YouTube video and it should play and we should be able to hear it. So I'll see if I can click on the video. If not, you'll just have to bear with and I'll try and get it up separately. So hopefully it'll play. There's an advert first, Leon, so we'll just ignore the advert and then we'll watch the video and you should be able to hear the sound. If you can't, I'll send you it separately.
Cheese.
An organism's genome is the entire genetic material of that organism, so the complete set of DNA. It provides all of the information required by the organism to function. DNA is a long chemical that contains bases represented by the letters A, T, G, and C. A gene is a section of DNA that codes for a particular protein. To learn more about DNA, watch this video. The human genome is 3 billion bases long, but only consists of about 20 to 25,000 genes. As an average sized gene is only about 3,000 bases long. This means that large parts of our DNA doesn't code for genes. The genome of every person on Earth is 99.9% the same. It's that tiny 0.1% that makes up genes that give us our unique differences. In fact, our genes are 98% similar to a chimpanzee's and 75% the same as a mouse. The Human Genome Project was an enormous project that saw scientists around the world collaborate to work out the sequences of bases in the human genome. The project started in 1990 and was completed by 2003, 2 years ahead of schedule. And here is the order of the bases: A, T, G, C, T. Only joking. If I was to read out the bases, one at a time, for 24 hours straight, it would take a whole century, and I certainly don't have time for that. So, now we have the information, how is it being used? Well, since the structure of DNA was determined in 1953, we have known that the order of the bases is very important, and that a change or mutation in this order can result in diseases. By sequencing the whole genome, scientists can use this information to work out the genetic causes of diseases, such as cancer, as well as developing treatments. The forms of genes, or alleles, associated with different inherited disorders have been mapped, which means that genetic tests can be carried out on people to see if they are sufferers or carriers of an inherited disorder. For example, a genetic test can tell if someone is a carrier of cystic fibrosis. By comparing the DNA of people living in different parts of the world, scientists have been able to trace human migrations. The Out of Africa theory, that the first humans left Africa within the last 100,000 years, has been supported by comparing the DNA of people living in different areas of the globe. The evidence shows which people are more closely related to others. Because of all the data generated by the Human Genome Project is available to anyone to view free of charge online, means that scientists around the world can use the information in their research. This helps to increase the number of uses for it. There have been major advancements in DNA sequencing since the Human Genome Project ended. It took 13 years and billions of dollars to sequence a whole genome during the project, but now scientists can do it in a matter of hours. And you can even get yourself sequenced for a small fee. These advancements will help develop new strategies for the prevention and treatment of disease. So, in this video, you've learnt what a human genome is, what the Human Genome Project was, and how the information from it
If you like the
is being
video,
used.
give it a thumbs up.
Right, so what I've got for you next after the video is some questions. I will read you the questions out, you can give them a go, and then I'll go through the answers real quick. So I'll just load them up, so again it might take a little bit of time. So the first question is, define what a genome is. So that's question 1. Question 2, in the 1990s, biologists started the Human Genome Project. What was the original aim of the Human Genome Project? The next question, so 2B, identify where scientists were able to obtain human DNA for analysis in the Human Genome Project. So that one starts with an N. Think about your cell. 2C, describe how the Human Genome Project has been extended. 2D, suggest a reason why the project was extended. 2E, describe the advantages of the scientists being organised into teams around the world to work on the Human Genome Project. And the last one, 2F, describe how scientists used genome sequencing techniques such as the Human Genome Project to establish a classification relationship between two species. So pause it here, give them a go back and forth if you need to in the video, and then I'll read out the answers. So I'll whiz through the answers, Leon, when I actually open up the correct one. So the answers, so for question 1A, oh, not sure what it's doing, hang on. Open, it's not opening, Leon, you're never going to have the answers. Right, we're finally there. So 1, the term genome means the entire genetic material of an organism. 2A, the original aim of the Human Genome Project was to sequence the entire human genome. 2B, scientists were able to obtain human DNA from all of the chromosomes in the nucleus and DNA in mitochondria. 2C, the Human Genome Project has been extended by initially sequencing the genomes of 1,000 people and now 1 million. 2D, the project was extended to find out more information about human DNA and to compare people. 2E, they were able to complete the project much quicker. It also spread the cost around the world by sharing it out. And 2F, by sequencing both organisms and comparing similarities. So in the video you had apes and humans. You were able to see similarities and differences, and the degree of similarity might suggest the degree of closeness in relationships. So give them a mark, see how you did, and you can move on to the next lesson when you're ready.